Hello. Thanks for the invitation for the organizers. So Moritz, Laura, thank you so much. And thank you so much for organizing this fantastic meeting. So my talk is visualizing the dominant GPCR coupling of GNAO1 mutations. Basically, you have seen this slide already. That is what we do in the lab of Vladimir Katanaev's. And I will be focusing mainly in this particularly dominant mutant function that has been described for G-alpha mutant. So basically that's what we call the dominant GPCR coupling. And this is what Henrik described this morning, is that the behavior of some of the mutants G-alpha to bind the activator receptor and trap it and in a so-called non-functional coupling to the receptor. Just because the explanation is so long, we decide to call it dominant GPCR coupling in our lab, so that to keep the name short. And as you have seen this morning, so these are more or less a summary of all of the tools we are using in the GPCR field to determine the coupling of the G protein to the receptor and the activation of the G protein. So basically, you can see the beta-gamma dissociation from G-alpha-O, the levels of free beta-gamma, and the direct interaction between the receptor and the G-alpha-O subunit, or also can be done indirectly by doing like the split nanolook that Kirill's lab does, using just the interaction between the beta-gamma or the beta with the receptor directly. So the main problem here is that to determine that the mutants has this dominant PCR coupling is that you basically need to take few of these assays, and only the combination of those will tell you, yeah, well, yes, this mutant seems to trap the receptors, seems to bind to the activator receptor and trap it. So that, if your goal is try to find like generate a tool for a platform for drug discovery or drug repositioning from FDA-approved drugs, these tools are not very helpful. So that's the reason our aim in the lab is try to generate a tool that will allow us to directly visualize the dominant GPCR coupling of the mutants in just one assay. And this is challenging because I was planning to use a microscope approach, so with cells. And if you want to visualize the GPCR, so what we are using in the lab is the muscarinic 2 receptor, which is a model GPCR from the GPCR's family that interact with GIO family. This is a GIO couple receptor. And when you express them in cells, the problem what you have is that the receptor accumulates intracellularly. So you can see here that barely you can see the plasma membrane localization of the receptor. And then if you immunostain well, in this case, you have a GFP that is extracellularly localized in the protein. So you can immunostain the GFP there, and you can clearly see that the receptor reaches the plasma membrane. But when you look at the GFP fluorescence, you see that it's accumulated everywhere. And the plasma reticulum, Golgi apparatus, you name it, it's everywhere. So on the other hand, when you express G-alpha-O in cells, and we are using the HEC293T cells, you clearly see that G-alpha-O is already readily detectable at the plasma membrane, because G-alpha-O is just meristylated and polymethylated, so lipid modifications target G-alpha-O directly through the plasma membrane. So based on this, what we decided to do basically was to take a rather old technique that is what's called the bimolecular fluorescent complementation, and we use it for the YFP, so for the yellow fluorescent protein. And in short, I will call it this now split YFP. And then what this technique is, okay, for the GPCR field, they are all familiarized with this. So basically it's that you take the yellow fluorescent protein, you cut it in two parts, you put one part in the receptor, you put the other part in the G-alpha subunits, and because these two proteins will be in close proximity mostly at the plasma membrane, so we speculate that the complementation of the yellow fluorescent protein, that is what will happen only at the plasma membrane or mostly at the plasma membrane, will allow us to see fluorescence of this complementation, of this complex, only at the plasma membrane. And basically, additional advantage that this system may have is that because this complementation of the fluorescent protein is irreversible. So you will force the receptor, because of the close proximity of this complex, to maybe bias the signaling towards the G-alpha-O that you are coupling to the receptor over endogenous G-alpha subunit that normally interfere with the measurements when you do this kind of assays in cells. So this was our idea, speculation, and well, this is how it looks like in cells, basically. So we succeed. So when you do the split YFP for the muscarinic 2 receptor and G-alpha-O, you clearly see that the fluorescence is at the plasma membrane on the cells, and you barely see some intracellular localization of the fluorescence. Most interestingly, what happens is when you stimulate the cells. So now we stimulate the cells with acetylcholine, and you see that this prominent plasma membrane localization just changes dramatically, and you see the appearance of many, many hundreds of vesicles in the cell. So the next step was to see, well, what happened with this split YFP when we analyzed the mutants. Now, we decided to take some of the most recurrent mutations in the patients, and we took two of the, what are basically the categories we have running at the moment. So we have two mutants that are from the epileptic, associated with the epileptic phenotype. We took two mutants that are associated with the movement disorder phenotype, another two mutants that are associated with this dystonia phenotype. So according to Henry classification of the groups that he designed, so this mutant is from the group 4, that is what he calls non-functional. These other three mutants are from the group 2, and I think these three mutant, one it is this cysteine that was not, this cysteine 215 was not analyzed in Henry's study, and this also was not analyzed, and this is this insertion of 2 amino acid that is this splice variant mutation. So now, and this is how they look like basically. So you stimulate the cells, all these mutants were able to complement the YFP, so we succeed to do the assay basically. But what you see is that the mutants that are associated with the mild phenotypes, with the dystonia phenotype, they just show no difference with the wild type. So you stimulate the cells, the complex endocytose, and you see massive amount of vesicle cell. But on the other hand, the other 4 mutants associated with these more severe phenotypes, they all seem to trap the receptor at the plasma membrane. So there is very, very few vesicles going into the cell. Now, this is a quantification. Basically, we quantify how much receptor is outside with or without stimulation. And as expected, you have just only significant differences for the wild type and the mutants associated with the dystonia phenotype. So now, to try to confirm this, okay, what we took a look is what are the next steps into the process of endocytosis of the receptor. And basically, once, as you know here, so the receptor, upon coupling, what is happening is the G protein dissociates, so alpha and beta gamma subunits separate, and then that opens up the pocket where the G-alpha subunit binds the receptor, and then it comes the GPCR kinases that can phosphorylate the receptors. Once the receptor is phosphorylated, what happens is that arrestin, beta-arrestin, that are molecules that are scaffold molecules that bind the receptors, and then all the machinery from the endocytosis can start, and the receptor can go for the endocytosis, basically. Now, what we speculate is that if the mutants trap the receptor at that level, so they bind the receptor and cannot decouple from the receptor, basically is the dominant negative, the dominant GPCR coupling, what we call. So we expect that the phosphorylation of the receptor should be blocked. And then we were lucky, we found an antibody that recognized the phosphorylated muscarinic 2 receptor, and that you can see the signal at the steady state with no stimulation is almost 0. And then when you stimulate the receptor, you see that basically all of these vesicles are now co-stained with the antibody that recognizes the phosphorylated protein. Now, what happened with the mutants? As you can expect, okay, only the mutants that are associated with the mild phenotype, the dystonia mutant, they produce vesicles that are phosphorylated, whereas all the other mutants, they have very, very low levels of signal with the phospho antibody. And that is what we quantify. So what we quantify here basically is we quantify the amount of vesicles that are contain the YFP complex that are phosphorylated. They have a positive signal for the phosphorylated antibody. So double label vesicles or vesicle-like structure. And as you can see, of course, as the dystonia phenotype, the dystonia mutants show no difference, whereas the other ones show very, very, very strong variants. So what we can tell so far is that, okay, the mutant associated to the most severe phenotypes, these 4 mutants, all of them, they just seems to trap the receptor at the plasma membrane. Well, now, we can immunoprecipitate this complex, okay, with an anti-GFP nanobody to confirm the phenotype. And basically, you have here 3 different species that you can see. You can see just the receptor alone, plus the complex, because you cannot separate this under these conditions. You have the complex YFP with the receptor G-alpha and the fluorescent protein. And the GPCR have a tendency to form dimers. So you also can see a dimer of this complex in the Western blot. Anyway, what you can see is that the antibody is very specific. If you do not stimulate, you have no signal for the phosphoantibody. Here is how much the protein was immunoprecipitated. Whereas when you stimulate, you find a massive signal for the phosphorylation. So the system works. And then when you look at the mutants, what we found is, as expected, the severe mutants block the phosphorylation of the receptor upon stimulation, whereas up to our surprise, that the mild dystonia mutants, they seem also to reduce, but modest, the 20 to 25% reduction in the phosphorylation, even though that they do not seem to affect endocytosis directly. So now, all of this was done using the split YFP between the muscarinic receptor and G-alpha. So now, since we are targeting the G-alpha internally, we decide to do this control using an untagged version of G-alpha. And what we did is we did the split YFP between the muscarinic receptor and the gamma subunits. And for this experiment, what we did was did 2 pools of cells that we transfect separately. One with the split YFP system for receptor and gamma subunit, with the co-expression of G-alpha, and other set of cells that we do not transfect with G-alpha. And then we co-culture them and we stimulate the cells. And this is probably the my favorite experiment because it's pretty nice. And then what we see here is that we see the endocytosis of the complex. We see the signal of the antibody. So we see that the vesicles are phosphorylated. And we see here in the magenta arrowhead, it's pointing to the cell that express G-alpha, versus the white arrowheads will point to cells that are not expressing G-alpha. So these cells do not express endogenous G-alpha. So what you have here basically is that the endogenous G-alpha is not there. So exogenously expressed wild-type G-alpha seems not to affect anything, the endocytosis and the phosphorylation of the receptor. But now what will happen when we apply these severe mutants, what we see is that all of them block the endocytosis seems to be blocked, the phosphorylation seems to be blocked, and this is the cell that expresses one of the epileptic mutant. Here is the next epileptic mutant that shows basically the same phenotype, so phosphorylation is gone, endocytosis is gone. Only in the cell that express the mutant versus the cells that do not express G-alpha seems to undergo endocytosis and phosphorylation just normally. These are the 4 severe mutants. And now, just the last control that we did is just to confirm this phenotype, what we do is just we use a construct that has no tag intracellularly. So because you have to avoid that this tagging of the proteins might interfere with the signal. So we use this GFP tag receptor with the G-alphas untagged, and we repeat the same experiment so we can immunoprecipitate the GFP receptor, and we found exactly the same phenotype. So what we can confirm here is that it seems that all of these severe mutants, when the receptor gets activated, the G protein that is formed by these mutants interact with the receptor, and then it blocks the receptor at the plasma membrane, and it doesn't allow for the rest of the machinery to come to the cell, to the receptor, to deactivate the receptor. And that is basically the model. This is a working model, so we still don't know exactly how this is work, but We have these two scenarios. There might be more scenarios possible. So this is exactly what the cryo-AEM structure that Henrik showed, that is this model of the open conformation of the trimetic complex that trap the receptor. Alternatively, we can envision that the mutants can separate beta-gamma, and the monomeric open alpha subunit can trap the receptor in this conformation. But we can also not exclude that maybe this it also can block the receptor, just the trimeric complex is still in the closed conformation can do that. So this is coming back to what we do in the lab. Basically, what we are doing now is, or what we will do in the future is, now we know that this system can be used for screening, for repositioning of FDA-approved drugs, and we will just run all of this in the near future to screen this drug and see if we can revert this dominant GPCR coupling. And with that, I would like to thank, well, this is what we are, and a special thank you to Massimiliano and the Familia GNAO1 that are funding this project that makes our life a lot easier. So